Hello students, we are discussing chapter 8, that is getting to know our plants. In this topic we are discussing now the leaf. In our previous videos of leaf, what we have discussed is what is the leaf. It is a part which synthesizes food for the plants, that is referred as a leaf. It is a flat green structure which has a wider base known as lamina or leaf blade. The boundary of a leaf is known as margins. Leaf has certain veins in it, and on the basis of these veins, leaves can be differentiated into two forms on the basis of their venation. That is the arrangement of the veins over the leaf blade or lamina. The first one is reticulate venation and other one is parallel venation. The venation in which the veins are present in a network like in a net-like structure are known as reticulate venation. Whereas the venation, the arrangement in which the veins are arranged parallelly to the midrib or parallelly to each other and perpendicularly to the midrib, that is known as parallel venation. There is a relationship between root system and venation. That relationship says that if a plant has a reticulate venation, then there are chances, the most probability is that plant has tap root system. अगर एक प्लांट के पास रेटिकुलेट वेनेशन है, then the most probably it has taproot system, and if a plant has parallel venations, then there are chances that it has a fibrous or adventitious root system. Now moving forward, we will discuss the functions of leaf. The first function of leaf is manufacturing of food. Leaves manufacture the food. As they manufacture the food, the leaves are referred as kitchen of the plant. They are referred as kitchen or the food factories of the plant. I will discuss it why in detail. What plants have is, plant have leaves and leaves have a green color pigment known as chlorophyll. Plant cell has a cell organelle. You will study it in your higher classes, bache. That is known as chloroplast. It is present inside the cell. That chloroplast actually have that green color pigment. This is green color pigment, bache. That is known as chlorophyll. What is special about this pigment? It has the tendency, it has the capacity to hold, to absorb, to trap the sunlight. सनलाइट को रोकने की, उसे ट्रैप करने की कैपेसिटी है क्लोरोफिल के पास. एंड बाय दैट सनलाइट प्लांट्स कैन कन्वर्ट कार्बन डाइऑक्साइड which they take from atmosphere, water which they take from the soil through roots. Water and carbon dioxide in the presence of sunlight are converted into a food, that is glucose. Glucose is an organic molecule. Along with that glucose, plants also release oxygen gas, the life supporting gas for the animals. So this process of formation of glucose with the release of oxygen from carbon dioxide and water in the presence of sunlight in chlorophyll is known as photosynthesis. All things are related, bache. What is photosynthesis? The production of glucose, that is food, and the release of oxygen. इनका प्रोडक्शन करना बच्चे और ऑक्सीजन को छोड़ना. किससे? फ्रॉम कार्बन डाइऑक्साइड एंड वाटर. कहां पर? इन क्लोरोफिल ऑफ लीव्स इन द प्रेजेंस ऑफ सनलाइट. दैट प्रोसेस इज नोन एज photosynthesis. Photo means light, बच्चे, and synthesis means putting together, to synthesize, to make something new. इन्हें दोनों को साथ रखकर क्लोरोफिल के अंदर सनलाइट की प्रेजेंस में हम क्या बना रहे हैं? ग्लूकोज. मतलब हम नहीं, प्लांट्स बना रहे हैं ऑब्वियसली. और क्या रिलीज कर रहे हैं बच्चे? ऑक्सीजन गैस. दिस प्रोसेस ऑफ फॉर्मेशन और मेकिंग फूड बाय ग्रीन प्लांट्स फ्रॉम कार्बन डाइऑक्साइड, water, and in the presence of sunlight or by the use of sunlight release or results in the formation of glucose with the release or along with the release of oxygen gas is known as photosynthesis. So that's why we say that plants manufacture the food. they use carbon dioxide and water in the presence of sunlight, in the chlorophyll of leaves, or in the presence of chlorophyll of leaves. They synthesize glucose and release oxygen gas along with it. And this process is known as photosynthesis. A very, very important process for your biology, for your science bache in your higher classes as well. As bache, plants keep on performing the photosynthesis whole day. Whole day sunlight is available and the plants keep on performing the photosynthesis. So glucose forms in excess bache. Glucose बनती जा रही है. Some portion of that glucose is used by plants to produce energy. Plants भी तो activities perform कर रहे हैं ना बच्चे. Plants grow कर रहे हैं, plants fruits बनाते हैं, plants flowers बनाते हैं. इसमें बच्चे energy जा रही है. For that energy they use some part of the glucose. And the rest extra glucose, जो बच गया glucose, that is stored in the form of starch. So if someone asks you what is the reserve food, reserve मतलब stored, जो हमने store करके रखा हुआ, reserve करके रखा हुआ, सुरक्षित तरीके से रखा हुआ, ताकि हम उसे later time में use कर सके, hard times में use कर सके, that is known as reserve food. So what is reserve food in case of plants? Starch. Basically starch क्या है बेटा? ग्लूकोज एक छोटे स्ट्रक्चर और स्टार्च एक बड़ी स्ट्रक्चर है. ग्लूकोज 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 जुड़कर क्या बनाते हैं? स्टार्च बनाते हैं. मल्टीपल यूनिट्स ऑफ ग्लूकोज जॉइन टुगेदर टू फॉर्म अ लार्ज स्ट्रक्चर नेम्ड एज स्टार्च. एंड स्टार्च इज द रिजर्व फूड ऑफ प्लांट्स. नेक्स्ट बच्चे, as we can discuss the whole process that plants can synthesize food by the help of sunlight. That is why plants or leaves of the plants specifically are known as food factories or kitchen of the plant. Kitchen होता है ना बच्चे जहां खाना बनता है. तो plants में खाना कहां बन रहा है? Leaves में. Photosynthesis कहां हो रही है? Leaves में. सिर्फ leaves में क्यों हो रही है? क्योंकि chlorophyll is present in the leaves. That's why leaves of the plants are referred as food factories, the factories where food is synthesized, or kitchen of the plant. Now we will discuss an activity. By that activity, we will confirm that starch is produced during photosynthesis. That after photosynthesis, plants' leaves store their extra food, that is glucose, in the form of starch. Before studying that activity, bache, before discussion of that activity, first we'll discuss how we will check for the presence of starch. To check the presence of starch in a sample. Bache, about this test we had studied in components of food, I think. There's a chapter 2 in your book. In that we had discussed how to check the presence of starch. To check the presence of starch in a sample, pour iodine solution over it. If starch is present, then the purple color iodine solution changes its color to blue-black. Confirms the presence of starch. बच्चे कैसे पता चलता है स्टार्च किसी चीज में है या नहीं? अगर किसी चीज का, किसी सैंपल का टेस्ट करना चाहते हैं, स्टार्च टेस्ट, then pour iodine solution over it. The original color of iodine is purple color. But when we pour an iodine solution over a starch containing substance like potato, sweet potato, that will be converted into blue-black. उसका कलर क्या हो जाएगा? Blue-black. So the color of iodine solution changes to blue-black in the presence of starch. That is the confirmation test for starch in any sample. समझ रहे हैं बच्चे? तो हमें स्टार्च चेक करनी है तो हमें क्या करना पड़ेगा? आयोडीन सॉल्यूशन डालना पड़ेगा. Now बच्चे will discuss the activity. The activity is named to show that green leaves produce or stored food in the form of starch during photosynthesis. कि leaves में photosynthesis के दौरान ultimately क्या बन जाता है? स्टार्च. पहले ग्लूकोज बनता है बच्चे. ग्लूकोज किस फॉर्म में स्टोर होता है? स्टार्च की फॉर्म. So for that we require certain, various things, various devices, various instruments. Like we require a leaf, obviously, bachche. For that we have to go for the check beta. Then we require a tripod stand. It looks like a dipod stand. It has two legs, but actually it has three. The third one is present on the other side. We require a beaker. We require water. We require a Bunsen burner, a matchstick, test tube, a test tube holder, iodine solution, a dropper. This is wire gauge बच्चे. ये एक जाली होती है बच्चे जिसे रखते हैं ताकि ग्लास डायरेक्टली हीट ना हो. That is referred as wire gauge. That is present in your laboratories बच्चे. One more thing we require in this experiment, that is alcohol. What alcohol do is, alcohol removes all the chlorophyll of the leaf. One precaution also we must have to keep in mind, that we do not heat alcohol containing test tube or beaker directly. जिस बीकर जिस टेस्ट ट्यूब में हमने अल्कोहल डाला है हम उसे डायरेक्टली फ्लेम पर नहीं रख सकते. Why? Because alcohol is highly flammable. बहुत जल्दी आग पकड़ता है बच्चे. It catches fire very easily. That's why it is referred as highly flammable. जो बहुत जल्दी से फ्लेम में कन्वर्ट हो जाए, जो जल्दी से पकड़ दे आपको. That's why never ever burn or not burn, heat the container which has alcohol directly. So in this experiment, the first step in this, the method, the first step is take a leaf बच्चे. A green leaf from a plant. Then, in the second step, boil the leaf for 10 minutes in water. That's a beaker बच्चे. It contains water. It has leaf in it. This is wire gauge. This is a tripod stand, and we are heating it by the help of Bunsen burner. These you have to represent only in one diagram. That is enough for you. So that is, after heating it, we keep it out. After 10 minutes, we make it dry a bit, and then keep it in a test tube. This is test tube. This holder is test tube holder to hold that tube बच्चे, ताकि हम उसे डायरेक्टली ना पकड़े. अगर हम उसे डायरेक्टली पकड़ेंगे तो वो गर्म होगी तो हमारा हाथ जल जाएगा. This contain alcohol. What alcohol do is, alcohol removes all the chlorophyll from the leaf. It will make the leaf decolorize. Next step is heat the leaf or boil the leaf in alcohol for 10 minutes. 10 to 15 minutes are enough बच्चे. That will make the leaf colorless. After that, wash the leaf with water. This leaf is colorless here. यहाँ वो लीफ क्या हो चुका है? कलरलेस. Why? Because when we heat it in alcohol, alcohol removes all the chlorophyll, and due to presence of chlorophyll, the leaves are green in color. If we remove that green color pigment, the leaf become colorless. Then after, why we are then washing it with water? So that if any chlorophyll will be remaining there, that will be also removed. अगर थोड़ी बहुत chlorophyll बच गई है तो पानी उसे remove कर देगा. Then in the last step, pour few drops of iodine solution. बच्चे यही तो डालना है ना. चेक कैसे करेंगे स्टार्च बना था या नहीं? इस ग्रीन लीफ में स्टार्च बना था या नहीं, कैसे चेक करेंगे? आयोडीन सॉल्यूशन डालेंगे बच्चे. This is iodine solution. We are pouring it by the help of a dropper. This is a petri dish. It's a glass dish in which we place the leaf. We'll observe that this leaf will changes its color to blue-black. ये जो leaf है बच्चे, जो colorless था पहले, क्या हो जाएगा? Blue-black हो जाएगा. और बच्चे हमने अभी किया, if iodine solution's color changes to blue-black over a sample, then that will confirm the presence of starch. अगर आयोडीन का कलर पर्पल से ब्लू ब्लैक हो गया तो क्या होगा बच्चे? दैट विल कंफर्म द प्रेजेंस ऑफ स्टार्च। तो बच्चे इस लीफ में जैसे ही हमने आयोडीन सॉल्यूशन डाला है, हम डालेंगे, दैट विल बी चेंज्ड इट्स कलर टू ब्लू ब्लैक। दैट विल कंफर्म द प्रेजेंस ऑफ स्टार्च इन इट। I am again repeating you or explaining you this activity, बच्चे. First of all, we have to take a leaf, a green leaf. Then we boil that leaf for 10 minutes in hot water. After that, we place that leaf, we keep that leaf in a test tube and heat that test tube. The test tube contains alcohol by placing it in a beaker containing water. Do not directly heat the alcohol because alcohol is highly flammable and it catches fire very, very easily. So to prevent any accident, do not directly heat the alcohol or alcohol containing container. Test tube. What does alcohol do? What is the function of alcohol? Alcohol removes the chlorophyll from the leaf. So the leaf will become colorless. Then that colorless leaf is washed with the water so that the remaining chlorophyll, whatsoever the small portion of chlorophyll is left there, is also removed. Then we make it dry, and then over that dry colorless leaf pour few drops of iodine so that we can get a check that whether starch is present or not. And if the purple color of iodine solution changes to blue black, that will confirm the presence of starch in it. अगर बच्चे आयोडीन का सॉल्यूशन का कलर ब्लू ब्लैक हो गया तो क्या कंफर्म करता है? स्टार्च। और हम जब इस एक्सपेरिमेंट में ऐसा करेंगे, when we pour iodine solution over this colorless leaf, the color is changed. That means starch is present in that leaf. That means the starch is formed during photosynthesis. इस बात को प्रमाणित करेगा, इस बात को फिक्स करेगा, सर्टेन कर देगा कि फोटोसिंथेसिस में स्टार्च फॉर्म होता है. So the conclusion of this activity is that starch is formed during photosynthesis.